Great. Okay, so thank you very much, Sally, for your introduction, and thank you very much to everyone for joining me this evening. I'm very excited to give you a talk on the work we've been doing in the Vasse-Wonnerup estuaries over the last 5 years that are aimed at trying to both understand and improve water quality within the estuaries. So let me make sure I can actually move my slides. Sorry. Oh, hang on. There we go. Hopefully that will continue. So the Vasse-Wonnerup estuaries, as most of you know, are two beautiful estuaries located close to the city of Busselton, about 2.5 hours south of Perth in Western Australia. And these estuaries are broad, shallow estuaries. The Vasse estuary is about 7 kilometres long, and Wonnerup's fairly similar. And these estuaries terminate towards the coast in long, skinny channels. These estuaries are highly valued by both the local community and internationally. And here are just some pictures to show you why they're such beautiful and valued wetlands or estuaries. And probably the most obvious is they support a really large number of birds at particular types of the year, especially in December and January. And so high that in December and January, the Vasse-Wonnerup wetlands have the highest density of waterbirds in Western Australia. And because of those really high numbers, the wetlands have Ramsar status, which means they're recognised by international conventions for their importance for waterbird habitat. On a more local note, they also support the largest breeding colony of our state emblem, the black swans. So highly valued, really beautiful wetlands, but just like other estuaries around the world, they really do show symptoms of suffering under the pressure of a growing human population. And here are some pictures from the Vasse-Wonnerup estuaries, where you can see on the lower left panel, we have discoloured water, thick green, smelly water, and that's because of high concentrations of algae or phytoplankton. These are small single-celled organisms that grow in abundance when the concentration of nutrients is high. And sometimes those phytoplankton produce toxins as well. And so they're generally referred to as harmful algal blooms. We also have conditions in the estuary where rather than phytoplankton blooms, we have macroalgal blooms. And that's in the upper right-hand corner. We have thick accumulations of what looks like weed growing. And both the algal blooms and the macroalgal blooms are driven by high levels of nutrients. So the nutrients are typically nitrogen and phosphorus. They provide the food that these organisms need to grow. One of the problems with these organisms, as well as looking unsightly, is that when they die, they break down, and the breakdown consumes oxygen. And so as they break down, they strip oxygen from the water, and that makes it very hard for aquatic organisms to survive. The Vasse-Wonnerup has suffered from 7 fish kills in the last 10 years. You see on the right-hand side, which just shows that the estuary's water quality is inadequate for supporting healthy fish life at certain times. So this is really unfortunate set of symptoms, and the best way to try, as a management agency, to try and resolve some of these issues is to understand how the estuaries work. And so I want to give you a bit of an overview of that right now. So the Vasse-Wonnerup, I've been referring to them as wetlands or estuaries. They are estuaries in that they receive freshwater flows from rivers, and I'm showing the rivers here. Many of these rivers drain either urban or agricultural land, and so they tend to become nutrient enriched. And so these rivers deliver high levels of nutrients to these estuaries. The estuaries, the skinny channels at the end of the estuary join together towards the coast, and they flow, the water that enters the estuary flows out through a single ocean entrance that breaks through a sandbar at Wonnerup Inlet. So these two estuaries are exposed to the ocean through this relatively small channel through the sandbar. Now this sandbar, just like many other West Australian estuaries, can close seasonally, and so when that happens, the estuaries are actually isolated from the ocean. we also now mechanically open that Wonnerup Inlet ocean entrance if needed. So these estuaries are unique in that they share this single ocean entrance, but there are other factors that make them even more unique. And that's because they have surge barriers at both of their mouths. So just here's a beautiful aerial photo, and there's the ocean entrance down there. And then the Vasse surge barrier is very clear in the foreground of this photograph. These surge barriers are in place to prevent seawater from flowing into the estuaries, and I'll say more about why they're essential shortly. But they're the only permanent surge barriers on estuaries in Western Australia, so very unusual situation. And the community and scientists have long been aware that many of the water quality problems that I showed you in that previous slide often tend to focus around these surge barriers. And you can see that in this picture here. Upstream of the surge barrier, we have green pea soup-like water, and downstream you can actually see the bottom. The water's much clearer. So the surge barriers are definitely being implicated in potentially somewhat causing those poor water quality issues that we often have. And so as part of this program, we've investigated how the surge barriers are impacting water quality. Before I give you some of the results of that program, let me just say a little bit more about the surge barriers, because it's important to understand what they do. And so the surge barriers, they've been in place for a really long time. They were first installed in 1908 by the early settlers, and they were installed to largely prevent seawater from flooding adjacent agricultural land. Today, they're really important to protect Busselton and surrounding land from storm surge flooding. Busselton would flood under high ocean levels if it wasn't for the presence of these surge barriers. These surge barriers are relatively simple construction. They're one-way flow structures. They let fresh flooding waters out, as shown in this picture. But then when ocean levels rise, those gates that are open in this picture are pushed closed and seawater can no longer get in. The structures that are present today were updated and constructed in 2004. And in 2004, there'd already been many decades of poor water quality. And so the gates were designed with a few additional features that you probably won't find on any other surge barriers. And that is, they included a fish gate. The fish gate is just a small opening to allow fish. And the goal was to allow fish to move from poor quality estuary water downstream into the inlet to prevent fish kills. And there's also a propped gate that is just a larger gate that can be opened to allow seawater into the estuary. I'll also say this figure also shows some checkboards. Those checkboards are removed in winter and then added back in at the end of winter, and they act to hold fresh water back, so raise the freshwater levels within the estuary. Now, the management of these gates is governed by a partnership called the Vasse-Wonnerup Wetlands Partnership, and there is a group of management agencies, and they decide how that gate is managed. And those gates have been managed consistently since 1990. And the 1990 management guidelines that were developed have generated a very distinct annual pattern within the estuary. And so I want to tell you about that now. Before I do, I just point out, so I'm going to be talking about water levels next, and anyone can go and look at the water level in the estuary. On the edge of each surge barrier, there's a staff gauge, or it's just like a long ruler. You can read off the water level, or we don't go out and read it every day. We've had loggers installed in both estuaries for about the last 20 years to provide us with data. So I'll show you that data now. So here is a figure showing how the average water level varies in the Vasse estuary over the course of the year. So on the x-axis, I have time. It starts in July and ends in July, so it's a whole year of data. And on the y-axis is the Vasse water level. You could go on and read off that water level. The units are meters AHD. That's meters Australian height datum, where 0 meters AHD is sea level. So negative numbers mean below sea level, positive numbers mean above sea level. Now what I'm showing here is the black line is the average water level from our loggers over 1997 to 2014. It's about 20 years of data. And the reason I'm stopping in 2014 is because after 2014, we started changing the system. And so what I'm showing here is really how the estuary has been in the recent past. So the black line's the average, and then I'm shading in a purpley color to show the variation in levels that has been experienced over those years. So I hope what you can see early on in the graph, around here in July and August, we have high water levels. Also, there's a lot of variation within the shading, and that's because the water levels are very much influenced by the rainfall patterns that we get. But in general, we have high water levels associated with winter rainfall. To explain the rest of the graph, I'm going to shade it to make it a little bit easier. And so what we have is, as the water levels are high at the end of winter, in around about September, the checkboards are added to the surge barrier, holding water back. Then the estuary is basically isolated from seawater. Water can't get in or out. It's isolated from the ocean, sorry. Water can't get in or out. And so what happens is the estuaries just evaporate. And you can see that we have this very slow and steady decline in water level in the green section as evaporation drives a reduction in water level. Now, if that water level, or if that was allowed to continue through into summer, the estuary would evaporate down to about negative 0.4 meters. So that would be right at the bottom of the scale. And that would happen by March. And at negative 0.4 meters, the estuaries, both estuaries are almost completely dry. There's really just a little bit of water left in the deeper parts of the channels close to the ocean. So the gates were completely closed up until the 1980s. And at the end of that time, concerned community members sort of agitated to try and allow some seawater into the estuary so that the estuary wouldn't completely dry up. And this is where some 1990 guidelines were established by Water Corporation, where after some consultation, they decided that if the estuary reaches a level of negative 0.1 meters, the gates could be opened to allow seawater into the estuary, thereby preventing complete drying up. So that pink line shows where the water level target was over summer. And the gates were opened just to maintain the water level at negative 0.1. And so as you can see, that red section, it's relatively flat, and it's also nowhere near as variable as the rest of the year. And that's because that's when management and gate management was controlling the water levels. So it's really very distinct time periods and different management in each season. How do these water level variations impact the ecology? So I just have some pictures to show what happens at each time of year. So in upper left, during winter, the estuary is full. It's very beautiful. It's an important time for swans nesting. And it's also an important time for macrophytes, or the seagrass, to grow and flower. Then as the estuary level starts to decline into that green section, we start having mudflats appearing. And at the same time, the estuary receives a large number of visitors from international and resident migratory birds. And so we have a large number of birds arriving in the estuary, and the estuary is able to support those large number of birds because of the wide range of both mudflat as well as open water habitat. And so we get our highest bird numbers in December and January during that green period of time. Then as we head into summer, even with the current guidelines, much of the upper estuaries dry out, and we end up with dry mudflats, as you can see in that photo. Also during the low water level period of time is when we have the highest number of algal blooms and fish kills. So that's when all our negative sort of water quality events tend to happen. And so because this is happening at the same time as we're managing the gates, this is what, after community encouragement, caused the development of this program here that was funded under Revitalising Geographe Waterways, called the Vasse Seawater Inflow Trial. And the goal of this trial, which started at the end of 2014, was to see if can the management of the vast surge barriers be changed to improve water quality in the channel. And so we've been undertaking very definite studies since 2014 to try and answer this question. And we've done this by, in the last 6 years, trialing 4 different ways of letting seawater into the estuary. And we've trialed different ways in different years. And the ways of letting seawater into the estuary have varied from, in the first year, we just used the normal management, so letting water in at -0.1 meters. But we've also tried letting no seawater in, letting seawater in sporadically every week, and then also letting seawater in early. So a whole range of possibilities have been investigated. And so I'll tell you what we found shortly. Underpinning all of this has been very careful and intensive sampling. So before I show some results, I'd just like to acknowledge and thank all the samplers who've worked really hard collecting really high quality data. And here's a picture of the samplers in the channel, in their boat. They travel up the estuary, they lower a probe that you can see on the lower left there into the water to collect data such as temperature, salinity, and oxygen. They collect water samples that are sent to a certified laboratory to give us nutrients. They also send water samples to the Department of Water Phytoplankton Ecology Lab, and their phytoplankton ecologists count and identify the phytoplankton. It's a very comprehensive suite of analyses. So now I want to show you some key results from the early years that have really guided what we've done in later years of this trial. So I'm showing you some data from December 2015, and this was the year that we let seawater in weekly. And I'm going to show you data from just 24 hours after we first opened the gates to let seawater in. And the data I'm showing you is salinity data. Salinity is how salty the water is. And I've plotted, and I'm using a color scale to show that. And so on this graph that I'm showing you, on the x-axis is distance up the channel. 0 is the surge barrier. And then as we head to the right, we're heading up and along the channel for up to 1 kilometer. And then left of the surge barrier is the inlet. And so left of the surge barrier, the water's dark blue, 36 parts per thousand. That's seawater salinity. Then as you go to the right and you go over the surge barrier, we get into lower salinity levels. And that's because we've just recently opened the gates. And so the salinity is not yet at seawater salinity. What we see is that the contours that I've plotted, so I've joined lines of points of equal salinity and generated contours. Well, you can see that the contours are all lying horizontal, and that we have within the estuary, we have deeper water has higher levels of salinity, and the salinity is lying in layers. And so this is called a salinity stratified system. And this isn't that surprising because we've just recently let seawater in. What is surprising is some of the other data that we've collected. So I'm now going to show you other contour plots, but of oxygen and temperature. So the top panel is the salinity I showed you. The middle panel is a contour plot of temperature. You can see there's not many lines. The temperature data is quite boring. The temperature is uniform everywhere. But then the oxygen data is we have a rainbow of colors. And so the oxygen data is very surprising to me because we have some regions of 0 oxygen. So that is the red. So good levels of oxygen at this time of year would be around 6 to 10 milligrams per liter. So if the system was healthy, it would have all been colored gray or light blue. But we have some areas of 0 oxygen, and 0 oxygen is lethal to aquatic organisms. Any aquatic organism that can't escape from 0 is going to die. And fish start to struggle at oxygen levels of below 4 milligrams per liter. And so the whole lower part of this vast channel is low in oxygen and would be detrimental to the health of the fish. So you can see that the oxygen is also formed in layers, and that's because that oxygen, the low oxygen, is being driven by the salinity stratification. Because of the salinity stratification, the deep waters are isolated from the atmosphere. And so those deep waters are no longer able to get oxygen replenished from the atmosphere. and so they're losing oxygen. The deep waters are also in contact with the sediments, and the sediments contain a lot of dead and decaying organic matter and algae, and as they decay, they consume oxygen, and not enough oxygen supplied from the atmosphere, so we end up having no oxygen in the bottom waters. So this is not a good situation. If that area of red water starts to grow, then we could potentially get a fish kill. So salinity stratification due to letting seawater in slowly is definitely not good for the water quality within the estuary. The following year, we had the opportunity to not let seawater in at all, and that's because the water level was fairly high. And so we kept the estuary fresh. And so here are the same figures, but for January 2017. And the upper plot is salinity. You can see blue below the surge barrier because it's marine salinity. But then above the surge barrier in the Vasse channel, there's no contour lines, and that's because the salinity is all a uniform 5 parts per thousand, relatively fresh. What is now surprising is that the temperature now is highly stratified. We have lots of horizontal lines of temperature. The water goes from 32 at the surface to 24 degrees at the bottom. And we think that temperature stratification developed because having the gates closed means the estuary is just sitting there baking in the sun, and it gets really hot. Now, temperature stratification is just like salinity stratification. It leads to isolation of the bottom waters. And so what you see with the oxygen is we've developed very intensive low oxygen regions within the Vasse channel. So, and it actually happens not letting any seawater in has actually generated worse low oxygen than letting a little bit of seawater in. You can see that the low oxygen area is actually extended all the way about 1.4 kilometers up the channel. So very not good conditions. So the low oxygen is very detrimental to aquatic life, but there's another reason we really don't like low oxygen, and that's because if we are to collect a water sample from those low oxygen regions and have them analyzed in the lab, then the water comes back with high nitrogen and high phosphorus, in particular ammonium and phosphate. These are the most bioavailable or most delicious nutrients for algal growth. And so when that water eventually gets mixed, it provides the perfect food to support phytoplankton bloom. And that's exactly what we see. This is a picture taken about a week after that contour plot the data for that contour plot was collected. And you can see, as I've already pointed out, we have very green water upstream of the surge barrier. The phytoplankton ecology unit investigated what that green color was, and it's from Anabanopsis arnoldii. That's a blue-green algal cell, and the concentrations reached almost 500,000 cells per mil. Incredibly high concentrations. That bloom spread all the way into the Vasse estuary, and you could see it on the Landsat satellites. So although it's caused by a microscopic organism, it got to such high concentrations you could see it from space. And that's all because it was fueled by this low oxygen areas that then generated nutrients that continued to fuel that bloom. So we've learned a few things in the first 3 years. I just want to summarize that. And so we learned that letting seawater in slowly creates stratification. And stratification is not good because that then leads to low oxygen in the bottom waters, and that drives nutrient release. The problem is, even when we don't let seawater in, the Vasse channel stratifies. It has a really strong tendency to stratify. We realize, minimize some of the stratification by letting seawater in quickly. And just another piece of information is the first algal blooms start in late November and early December. So guided by this learnings that we had from the first 3 years, we developed a new way of letting seawater in that we started in 2017, in the summer of 2017-18. And in this trial, we opened the gates up in early December with the goal to interrupt that first algal bloom. And by getting the salinity up very high, we're much less likely to have blue-green algae blooming. And they're the ones that have been causing us all those problems. in order to try and prevent salinity stratification from happening, we opened both gates when we opened the gates, and we modeled all these scenarios very carefully to make sure we didn't adversely flood any land or anything. And our modeling suggested that we couldn't keep both gates open all summer, but that we could keep the fish gate open all summer. And so we've kept the fish gate open all summer, and the goal of keeping that open is largely to have enough water movement to stop the temperature stratification from developing. And so we've done this early December seawater inflow for the last 3 years. And so I want to show you how we've changed the water quality since we've been doing that. So this is a slide where I've counted up the number of hours of low oxygen that we've had in the Vasse Channel. And I've just got each summer 1 to 6 listed over the top. And you can see from summers 1 to 3, we had, you know, up to 200 hours, many equivalent days of time when we had almost no oxygen in the bottom waters. Then summer 4, where we first allowed seawater in early December, the first summer we did that, we got no times where we had low oxygen. So we really thought we'd actually largely solved the system. And so that's why we've done the early seawater inflow in years 5 and 6 again, because things were looking really good. But we didn't, we certainly didn't solve it completely, because we did have some times where we had no oxygen in the bottom waters. But really the last 3 years we've had, we really have vastly reduced the number of hours of low oxygen that we've had. So we've improved the oxygen situation considerably. By improving the oxygen conditions significantly, we've reduced the amount of nutrients that are released from the sediments. And so that, in combination with the higher salinity water, has improved our phytoplankton cell situation. So here's a figure of the data from the phytoplankton counting. And on the x-axis is just time going from 2015 up to 2020. And I've shaded the summer in yellow just to make it a little bit easier to look at the graph. And on the y-axis is the number of cells per mil. So it's how many cells can be counted. And I'll note that the y-axis goes up to 500,000 cells. So these are, anything that's plotting up towards the top of that axis is a thick pea soup color. So you can see in the first 3 years, 2015, 16 and 17, we had a lot of red. Red is harmful phytoplankton species. And in the Vasse-Wonnerup, that tends to be blue-green algae. And the problem with blue-green algae is they produce toxins that can be very detrimental to human and animal health. Then I've also plotted in grey phytoplankton that are not harmful. So the first 3 years we had very high and persistent levels of harmful blue-green algae. Then the first year we let seawater in early, we had greatly reduced levels of phytoplankton, both harmful and non-harmful. And in the successive years, we've had very low levels of harmful algae, but we have had one pretty intensive bloom in 2019 of diatoms. Diatoms are another type of phytoplankton. They tend to be marine in nature, and they're much less hazardous than blue-green algae, and that's why they're called non-harmful. But at the level that we had in 2019, at 1.4 million cells, that level is definitely getting up to harmful to fish and other organisms. So we were lucky in 2019 not to have a fish kill. So because of that, let's have a look why we ended up with those high concentrations in 2019. And this just shows you that although letting seawater into the estuary early has improved the situation, we're really not in control of everything. And so let's look at what caused these 50 hours of low oxygen conditions. And it was from 2 events that happened over the course of the summer number 5. And the first event happened shortly after we opened the gate. We opened the gate, seawater came in, everything was looking great, but then a high pressure system came and sat over the Indian Ocean, and that has the effect of lowering seawater. And so the ocean level was very low, and so we weren't able to get seawater into the estuary. And so what we actually did was create the exact situation we were trying not to have, where we'd let a little bit of seawater in and then let it sit and fester. And that's exactly what it did. The seawater we'd let in stratified, we ended up with low oxygen, high nutrients, and the salinity was high enough that we didn't get blue-green algae, but we got diatoms, and they grew up to 1.4 million cells per mil. That if that situation had persisted, we very likely would have got a fish kill. Luckily, the high pressure system moved off, ocean level rose, we were able to get seawater in, and that bloom was flushed away. And so nothing happened. So we were very lucky there, but it shows you we're not in control. Ocean level is really important in the management of this system, and we don't have control over that. The second event happened at the end of the summer, and the sandbar closed. Water Corporation do a great job in keeping that sandbar open, but that also can be very difficult to keep open at times. And so having the sandbar closed led to the water stopping its movement, and that generated times of low oxygen as well. But overall, we have improved the water quality significantly within the Vasse Channel. Now, the Vasse Channel is really important from that it tends to have fish kills and algal blooms, but it's the rest of the estuary that really has the very important ecological values that we treasure about these estuaries. And so it's important to see how our management solutions for the Vasse Channel has impacted the rest of the estuary. So everything that I've been talking to you about so far has mainly been at this site, Vasse 1, the upper right-hand corner there. And so let's have a look at how we've changed conditions at some of these other sites within the Vasse estuary. And all of the estuary is impacted by having the fish gate open. The water level in autumn is about 10 centimetres higher than it used to be in the last 20 years. 10 centimetres doesn't sound like very much, but it's a very flat, shallow system, so it is actually quite important. So in the Vasse Channel, the salinity rise is now happening in early December, and that's about 3 to 4 weeks earlier than used to happen on average. As that salinity, the salt wedge moves up the estuary, it takes some time. So by the time we get to the mid-estuary, excuse me, the salinity rises about 2 to 3 weeks earlier than it used to. Some other significant changes in the mid-estuary. Previously, the mid-estuary used to get up to 100 parts per thousand by the end of summer. And that's because the water would just evaporate away, and we'd end up with salinities about 3 times the salinity of salt water. Now we have some seawater coming in, and so the last 3 years, the salinity has been about 65 parts per thousand. So it's still way higher than seawater, but not as intensively so. Probably some of the biggest changes are in the upper estuary. And the upper estuary, VW26, that would typically be dry after mid-January under the 1990 management guidelines. But in the last 3 years, that has stayed wet all summer at a salinity of about 70 parts per thousand, so 2 times seawater. So what we've done, we've raised the salinity earlier and kept the estuary wetter. And so the problem with this change is that it's all happening in December. And in December is when we have the peak of the bird numbers. And so it's very important to understand what are the ecological impacts of having this early seawater inflow. And so since 2017, as part of the Revitalising Geographe Waterways program, there's been seasonal sampling of fish, birds, benthic invertebrates, and seagrasses and macroalgae. And I recommend, if you haven't already seen them, the 2 previous webinars in this series were on fish and the seagrasses. So I recommend looking at those talks and as they are discussed in the previous webinars. And next month will be the talk on waterbirds. And so I encourage you to tune in next month, to find out about waterbirds. But it's very important that we understand how our changes in the gate management are impacting the ecology of the system. And so I'll just tell you what we found with waterbirds, and saying that Kim Williams, who will be giving the webinar next month, is going to say a lot more about waterbirds. But I just want to show you some of the challenges in managing this system. Here's the bird numbers over the last 5 years. And again, I've shaded the summer periods in yellow, and I've labeled how we had things managed. and we don't have any data in the first year. But what we have is a highly variable bird numbers. You can see we only had 3 years of very high bird numbers. In the summer of year 3, where we didn't let any seawater in, had very good bird numbers. Then the first year of seawater inflow, the bird numbers were not very good, so that caused us concern. But the Wonnerup bird numbers were also relatively low, so we don't know if those bird numbers are due to the seawater inflow. In summer of year 5, we had very good bird numbers at first, but once we opened the gates, the birds disappeared. So summer of year 5 caused us additional concern. And then summer of year 6, we were sufficiently concerned that the Vasse-Wonnerup Wetland Partnership, the group responsible with managing and making decisions about the gates for these wetlands, was sufficiently concerned that we decided to try a hybrid early seawater inflow, where we opened the gates early, but once the channel was at marine salinity, we closed the gates to try and slow the progression of salt water so that the estuary remained fresh over December. That didn't work. We were unable to slow the progression of salt water, but we did have a really good bird year with some of the highest bird numbers recorded for December. So over the course of the 3 years of early seawater, we've had some average, good and bad data. So it's still very unclear how the ecology is reacting to this early seawater inflow. And it might be several years until some effects on some of the other ecological components are actually observed. So just to conclude, I'd like to say we've gone from a situation in the Vasse Channel where we have a thick, green, stinking waterway to, as shown in January 2015, to a much more respectable waterway shown in January 2018. So we really have significantly improved the water quality in the Vasse Channel. Unfortunately, it's unclear what the ecological impacts of the rest of the estuary are. It's unclear how the ecology reacts to this early seawater that we've done in the Vasse Channel. So there may be a balancing act between improving channel water quality and estuary ecology. This coming summer, the Vasse-Wonnerup Wetland Partnership, which is a partnership between the agencies that are responsible for the wetlands, that's Water Corporation, City of Busselton, Department of Water and Environmental Regulation, and Department of Biodiversity, Conservation and Attractions. So there are managers from that group in the Vasse-Wonnerup Wetland Partnership. They're responsible for taking into account all of the information, and they decide how the gates are to be managed. They have decided, following advice from the Science Advisory Group, to have another summer of early seawater inflow. So we gain the benefits of the improved channel water quality, but we're going to continue to collect ecological data so that if we show signs of detrimental impacts to the ecology, we can alter our gate management as needed. So that's all I want to say. I would just like to acknowledge there's been a huge number of people involved in developing, then collecting the data and analysing the data. So I'd like to acknowledge all the people in the Busselton and Bunbury and Perth Department of Water and Environmental Regulation who've done work on this, and also to thank the Science Advisory Group, who are scientists from Murdoch University, Edith Cowan University, Department of Biodiversity, Conservation and Attractions, and Otelia Ecology, who provide their time to provide advice on the science and the ecology of this system. And that allows us to have the best science and the data to help improve the management of this system.